Hi everyone, today topic is principle of ELISA. First, like and subscribe my YouTube channel. First, principle of ELISA. ELISA works on the principle that specific antibodies bind the target antigen and detect the presence and quantity of antigen binding. In order to increase the sensitivity and precision of the assay, the plate must be coated with antibodies with high affinity. ELISA can provide a useful measurement of antigen-antibody concentration. ELISA procedure. Next, ELISA procedure. ELISA is one of the easiest blood tests that can be carried out. It is rapid, quick, and requires a blood sample of the patient. The entire procedure of ELISA is mentioned below. An antibody is attached to a polystyrene plate, which is a solid surface, and is attracted or has an affinity towards bacteria, other antibodies, and hormones. A microtiter coated with antigen is filled with this antigen-antibody mixture, after which free antibodies are removed by washing. A second antibody specific to primary antibody is added, which is usually conjugated with an enzyme. Free enzyme-linked secondary antibodies are removed by washing the plate. Finally, the substrate is added. The substrate is converted by the enzyme to form a colored product, which can be measured by spectrophotometry. HCG protein, which indicates pregnancy, is detected by ELISA. A combination of blood or urine sample and purified HCG linked to an enzyme is added to the system. If HCG is absent in the test sample, then only the linked enzyme binds to the solid surface. The more the substance of interest is present, the more reaction takes place, and less of linked enzyme binds to the solid surface. These reactions are indicated usually with a change in the color of solution. Diseases that can be diagnosed using ELISA. ELISA can be used to detect some of these conditions: Ebola, pernicious anemia, AIDS, rotavirus, Lyme disease, syphilis, toxoplasmosis, Zika virus, carcinoma of the epithelial cells. Next, advantages of ELISA. Following are some advantages of ELISA technique. Results fetched from ELISA gives an accurate diagnosis of a particular disease since two antibodies are used. Can be carried out for complex samples as the antigen is not required to get purified to detect it. It is highly responsive since direct and indirect analysis methods can be carried out. It is a rapid test, yields results quickly. Possible detection for ELISA ranges from the quantitative, semi-quantitative, standard curve, qualitative, calibration curve models, extras. Easier to perform and uncomplicated process as compared to other assays which require the presence of radioactive materials. Next, applications of ELISA. The applications of ELISA are discussed below. The presence of antibodies and antigens in a sample can be determined. It is used in the food industry to detect any food allergens present. To determine the concentration of serum antibody in a virus test. During a disease outbreak, to evaluate the spread of the disease. For example, during recent COVID-19 outbreak, rapid testing kits are being used to determine presence of antibodies in the blood sample. For more information, So it was all about ELISA applications and its procedure extras.